on who we are. Matthew Fensky will be the next person to speak. He is our business development manager here at Cherney and has been leading our education team for quite some time. Lauren Ellie will be monitoring your questions and answers. She is our technical representative from our sales team and will be answering any questions that you guys may have. And I am Robert Brown, the client services manager here, and I will be doing some of the slides as well. So to get things started, here is Matthew Fensky.
Thank you, Robert. As Robert had mentioned, we do have some different interaction things. So what we'd like to have you start with right now is answering what industry are you calling in from today, just so we can kind of tailor up where we're talking, our discussion points, and give you a chance to do some polling. So I see some dairy and powders coming into play here. A lot of the other categories. So if you guys in the other category want to send into the chat function where your industry lies, we can help drive to that as well. So we'll leave that up in the background here a little bit. Actually, I'm going to close that out, but thank you for your interaction. And we'll get going with the presentation. So really the meat and bones of what we start talking today is our environmental monitoring program. What is it? Where do you come into play with it? And why in the world does it matter to you, to the consumer, and your customers at the end? And ultimately, it's all about food safety. When we start talking about environmental monitoring, we're really looking as a verification activity and risk mitigation. So we want to know where is the biological hazards in our environment, and we want to know, are we controlling that? So sanitation is a preventive control within the food safety plan. We have to have some effort of monitoring around that. And this environmental monitoring is part of that. Where we're going to spend most of our time today is understanding, as a technician, what do I do? What is my role in this, and how do I help impact the overall food safety of our product? So obviously there's been some inadequate events around this. We'll just leave these pictures on the screen and you can make your own assumptions off of what we're discussing there. But it's still this big global issue. So in 2011, the CDC released a few guidelines about how many foodborne illnesses are there? What is the impact of what you guys are doing every single day at your facilities? And what they found is there's still an estimated in the US alone, 48 million cases related to foodborne illnesses every single year. Of that 48 million, 128,000 people get hospitalized, and there's still around 3,000 deaths. So the efforts that you're putting forward from the 2011 numbers, I hope when they release them again, hopefully here in the next year, these numbers will be much, much smaller. And that is all thanks to you guys. And what we want to do today is really help give you the tools to continue to drive these numbers down. Part of this is understanding how quickly it can happen. So what we have on the screen now is a coliform example. And in one cell of coliform, if given the right conditions and unmanaged, it will replicate and duplicate itself every single 20 minutes. That means in one 8-hour shift that you're working, one single cell can become 16 million cells of coliform. Now, if we expand that out, Salmonella, as we'll talk about a little bit later, the infectious dose, how many cells it takes to get someone sick, can be as low as 1. So when we look at that 16 million number, obviously we start talking that this can happen very, very quickly. We can start with a clean slate in the morning and by the end have a higher risk. So that's why your sanitation and this monitoring really plays a key role. When I said ideal conditions, there's a lot of things that are going to come into play. So time and temperature, oxygen, pH, moisture, water activity, as you can see on the screen there. And a lot of you may be doing something called an APC count in-house or aerobic plate count. So we see that large number in this big group of organisms, and it's all dependent on oxygen. APC, the bacteria that are in there are only going to grow in the presence of oxygen, which is going to be a large amount of them. The other thing that I want to point out on here is we have moisture and water activity. These terms get confused very quickly, so I just want to spend a couple seconds to describe that moisture is all the liquid, whether it's bound to something or not within your product. Water activity is looking at how much of that is free. How much can a microorganism go and grab onto and use for growth? And that is looked at a scale from 0 to 1. So all these are going to play a factor into what microorganism can grow in our product, in our environment. And these are also going to be the factors that we're going to look for when we start investigating sites to see where they come into play. So when we talk about our microorganisms, we really want to think about where they're coming from. Now, if I exclude the raw ingredients up there, every single one of these things is going to be found within your manufacturing environment. When I say that, and we talk about environmental monitoring, that means our sources, other than raw ingredients, are all coming from the environment. This is why it's critical that we're really focusing on the environment and making sure that's clean, that we control that before it ever impacts our product. I have humans up there. I'm sorry, but we as humans, I took a shower this morning. I'm still microbiologically disgusting. And this is really going to come into account of, we want to keep our products safe from us, but when we talk about how we sample later, we're going to see an example of pushing the bag against your body to close it. We want to make sure that as a human, we're not negatively impacting our sample that we're sending off for testing either. So we really have two different groups of organisms that we can look at. The first is really going to tell us, is my environment sustainable for the growth of these organisms? And that's when we start talking about indicator organisms. Largely, this is going to be used in lieu of the pathogens in zone 1s and elsewhere within our facility. But you can see that we have coliform groups, Enterobacteriaceae, or maybe you hear it as EB, that really indicate to us, hey, my environment's right for the growth of Salmonella without actually testing for the Salmonella. So this is really going to give that overall risk of what our environment looks like. With that, we have our indicator versus pathogen, and you can see that in this case, a lot of times the US used to test for coliforms, still is testing for that. The European Union is changing over, or has changed over, and pushing us to do the same, to go to Enterobacteriaceae. And you can see that coliforms are within that family of Enterobacteriaceae. The other thing that indicator organism actually shows us is that some of our pathogens are showing up in there as well. So you can see our E. coli, our Salmonella. And that's something to be a key factor when we talk about how do we test in zone 1, which we'll describe later on. And really, indicators versus pathogen, we can look at a couple of different things here of how we're determining our results. And Robert's going to talk to us here in depth about that. But really, indicator, we're getting a number, we can set an action limit and track that over time, that number of cells of bacteria colonies that are there, and take action on it before it becomes a major issue. Pathogens, we're doing more of a seek and destroy. It's either present or it's absent. So Robert, I'll let you take over here and tell us a little bit more.
So like Matt had said, we have these two types of sampling and testing, the one being quantitative and the other one being qualitative. So on that quantitative, we're going to be using these for the indicator organisms. We're going to collect a consistent sample. Remember, we want the results to be the same for the same area. So the size and how we're sampling should always be consistent. Once we receive that, a portion of the sample is going to be incubated. Some sort of a plating test will be done, and then your result that you receive will come back as a number. And the units of measure after the number will be per gram, per milliliter, or per sample area. An example of that might be 150 CFUs per gram. When you get that data, you can trend that data over time. But the other side of it is qualitative. So qualitative is going to be, is it there or not? So we're going to use these for pathogens. And remember, you can collect a representative sample. So if you're going to sample, let's say a table or a leg or a drain, you can go ahead and swab as if you're looking for that pathogen. So you're going to get in there and really look for it, seek and destroy that area. Remember, the entire sample is incubated. It's going to go through the enrichment here, and your result when you get it back from the lab is going to be either presence and absence, or positive or negative. Units are going to be in the original size that we receive, like a 25 gram, a 375 gram, or per sampled area. So an example you may get will be negative per the 25 gram, or more commonly in the environmental monitoring would be negative per the sampled area. And then when you're going to trend it in your plant, you can go ahead and trend that by hits, by spots. So, different rooms, different areas, different zones where you're getting those hits that are positive. Now, when you send in your samples, the choice that you can make is to composite those environmental samples or to not composite. Compositing will give you an obvious cost advantage, and also maybe allow you to do more sites for the same cost. So when should we composite? So anytime that we're doing something that's presence absence, that's that yes or no, that positive negative, right? Anytime we're doing pathogens, things that you think are routinely negative or what you would consider to be clean, it's probably going to come back clean. I'm going to go ahead and throw it in a composite. Remember, when you composite, it's up to 5 sites, and those maintain the individuality. So if there is a presumptive, we can go ahead and quickly find out which one of the sites caused that issue. Now, when should we not composite? Something that we're going to do on a plate count, something that's an indicator organism that you're going to have trended data. You're going to want that individual data to put into your system to trend. Anything that you think is historically has a lot of hits, or that you're expecting hits, or because remember, we have to go back and rerun those tests at the end to find out which one was bad. That will negate your cost savings. And then anything that you do that you're going to investigate, maybe a hot spot or some place that you've had positives, you want to run those individually as well. So this next screen is just going to be kind of a map on how that compositing works. So you can kind of see, you send in the 5 samples, we go ahead and incubate and enrich, and then put those together to run on the platform. And then if those are a positive within that sample, we would go ahead and quickly rerun. In a few hours, you would know which one was a positive in that sample.
So as we start looking at our environmental monitoring program, we need to take the mindset that the goal is really to find the pathogens in the environment. And then we can control that in the environment well before it ever hits our product and puts us at risk for a recall. To do that, we really need to understand what are those pathogens and where do they grow, what's their factors for growth. So we're going to take a moment to look at these two, Salmonella and Listeria monocytogenes, as our 2 common pathogens that we want to just dive in a little bit deeper based on the industries that were represented at the beginning. So Listeria monocytogenes is really our relatively new pathogen. It comes in a family which we call Listeria genus. Listeria genus is typically what we are going to look for in the environment, as Listeria monocytogenes, the pathogen, will grow in the same conditions as what the whole family, the whole genus, will grow. But the Listeria monocytogenes is kind of the wimpy kid of the family and would be the last one to grow there. So if we can find we have a presumptive positive for Listeria genus, we know our conditions are right for monocytogenes and can control it before it ever becomes an issue. So as I go through the ideal growth conditions or the ranges in which the Listeria can grow, I want you to take a moment and think about within your facility, do you have these conditions available? Where would these conditions be available? And then the optimal conditions is really where you want to go get the most bang for your buck and take the actual sample. When I look at this and talk about temperature, optimal temperature obviously is at that 86 degrees Fahrenheit, but you can see there it grows all the way down to 33 degrees Fahrenheit. And actually, if you think about some famous recalls, it has been found in frozen products surviving. Big ranges on the pH, big ranges on the temperature. interesting part of this is when we look at those of you in the dairy industry and we talk, or other brine type industries, the amount of salt that it can survive. So 20 to 30% salt. Obviously our brine already has the moisture present for its growth. As that brine gets diluted out or gets washed out, now we introduce that risk for Listeria where we thought, hey, the salt is controlling it in the past. So Listeria obviously is kind of that headline of late within the food industry, and we have a lot of those high risk foods that are associated with it, but it can be across all kinds of different industries. So ice cream, caramel apples, cheese, lettuce, deli meats, just to name a few, are all areas where we've seen large outbreaks. Now, again, I mentioned at the beginning, this is one of the new pathogens, relatively speaking. So that's a constant learning of how is it mutating, how is it changing, and what other industries are actually impacted by it. Now, the one that we have had for quite a while and know is Salmonella. This has been over 100 years that we've known about Salmonella, and it's still causing billions of dollars in the industry for recalls and disposal of product. When we talk about Salmonella, there's only 2 species, but it has a ton of diversity within those species, where there's over 2,500 different serovars, all of which we consider pathogenic. So when we say we're testing for Salmonella, we're looking at all those serovars to understand, is it there, is it not there, because all those, like I said, we consider pathogenic. Looking back at our indicator organisms, I just wanted to throw this out. If you're testing for coliform, Salmonella falls outside of that category. Still going to have similar growth conditions, but not exactly. And that's where this push for Enterobacteriaceae, or EB, as an indicator has really become prevalent within our industries. When we talk about the growth conditions, a couple of things I want to point here is 98.6 degrees Fahrenheit. Now, I don't know about you guys, but I've had to take my temperature coming into the facility every day for the last few months. And it's typically right about that 98.6. So congratulations to everyone as you're taking your temp. You're perfect incubators for Salmonella. Obviously, within our industries and the environment, that's also a temperature that we may see maybe in our dryers and whey powders, or some of these drier ingredients where we don't have temp controlled environments. Again, that large range for the pH, large range for the temperature, ideally about 0.93 or higher for water activity, again 0 to 1, but can survive all the way down to 0.2. So you might know a few famous peanut butter recalls that this would have been an impact because of that low water activity. Looking at who was dialing in this morning, another thing to think about is whey powders. Obviously, those powders have a very low water activity as well.
Okay, so Matt talked about what we're looking for and where we're going to look for it. So now we're going to go ahead and talk about how we're going to go and take those samples and get them to the lab. So things to think about as we're building this EM program and as we're doing the swabs is, remember, we're verifying the effectiveness of the frequencies and when we're doing them, not so much the cleaning itself. We know that the cleaning will work, so we're just going to verify how often we're doing it, maybe who's doing it, when we're doing it, stuff like that. So we're going to also measure the effectiveness of the sanitary design, your personnel practices, your GMPs, right, and your operational methods. Look at the presence of pathogens and level of indicator and spoilage organisms. Discover those environmental sources, so maybe construction or things going on outside, people bringing stuff in. Assess equipment, utensils for harborage sites, and look for hotspots. And then lastly is looking for those trends. You know, where are we getting hits and how can we stop them from coming back in the future before they're a problem for our food.
I just want to jump in and apologize. I do see that we have a little thing blinking on the screen. I apologize for that interference. I will attempt to try and get that off as Robert continues here.
So the 3 areas that we're going to talk about sampling today are going to be your air, your water, and your surfaces. Within those surfaces, we're going to talk about the microorganisms, meaning the pathogens and the indicators as we spoke about, and then the metabolic byproducts, and then anything that would be a chemical residue. So the first thing we're going to talk about is the air. So what are we looking for in the air? We're going to look for the spoilage organisms or the indicator organisms. Why does this matter? Everybody has a heating system, a ventilation system, air conditioning, your HVAC systems. They're all important pieces and often forgot about, especially in the daily cleaning. So by doing this testing, we're going to test what air we're circulating throughout the plant and making sure it's not going to affect our food. So we want to test areas of product concern. So where do you have open product? Where do you have open packaging? Where could your stuff be exposed to the air when it's not sealed? Area where your sample device can be placed and remain relatively undisturbed. Remember, we want to test the true air, so we want those things to become undisturbed. Any area where outside air comes directly into your facility. So think about any doors that open, docks and dumpsters, overhead maintenance areas, stuff where your facility can be exposed to the outside world where things might be coming in. So just think about how do you pick your air sites. So there's two ways to grab those air samples. Active. So when you think about actively, we're going to pull that air onto media using an air sampler of sorts. And then there's passive, where we're literally letting the air to settle onto the media that we're going to test it on. The media is incubated and the colonies are going to be enumerated. Common things that we're going to see are going to be the APC count, the yeast and the mold. You may look for specific organisms for more troubleshooting. And then something else to think about that I often gets overlooked is your compressed air. So we're going to go test the plant air, the facility air. Are you using compressed air coming from your compressor, probably in a different area? So how are we testing that? So now Matt's got a short little video. We're going to talk about getting a proper air sample.
Being able to quickly and effectively test your air quality is an important part of maintaining a clean production environment. Routine air sampling will detect viable airborne particles and establish your baseline for future results. Results can be obtained through two options. One, a centrifugal air sampling device like the one Matt is holding here, and passive sampling. With an air sampling unit, a dish is placed inside the unit and a known amount of air is pulled through the unit and the sample collected. The sample is then sent to the lab for testing. This method will provide consistent amount of air for every test. However, it also has a upfront cost, as some plants may not have an air sampling unit. The second and less expensive option is passive sampling with a Petri dish. This Petri dish, like Matt is holding here, is the same one that you use in your lab and can be used for passive air sampling. So Matt has taken the Petri dish and will place it on a flat, smooth surface in the same environment that you are running production. Once the Petri dish is placed down, the lid is taken off to expose the dish to the air. Watch how Matt carefully places it on the ledge on the side with the bottom facing down. If the bottom is facing up, you have effectively doubled your surface area for testing. Remember, you only want to test what is in the Petri dish. So Matt will place it one edge on the side of the Petri dish and leave it there for 15 minutes. After 15 minutes, he will replace the cap and send it to the lab for testing. Note that when he took the lid off, he immediately put it on the side. He did not place it in his pocket. He did not walk around with it, and he did not place it on the bottom and place the dish on top of that. If you do this, you will effectively also test what is on the bottom of that Petri dish. So Matt now replaces the cap and takes it to his lab to be sent out for testing. The reason we do the air monitoring is once the results are received, we can change and effectively clean the environment if there is unwanted test results.
So after that little example, let's jump over to water. And really when we start talking about water and what we want to test for, we need to kind of know what we're using our water for. Is it going in as an ingredient? Is it just used for cleaning? Are we a dry environment and we're washing our hands when we're coming in the morning? All the way across the board, we want to make sure that obviously the microbial risk and that contamination risk is not part of it, but we may also want to take into account some chemical analysis on our water as well, especially in areas where we know we're on a private well and there's been issues with heavy metals, pesticides, other items that may be groundwater contamination. Ultimately, this is going to play into our GMP requirement that we're making safe water, that we're using the right water to clean, and not using something that's going to be an additional source for biological hazards or chemical hazards. So a couple of sites that we want to take a look at. Obviously, if we're using a water as an ingredient, we want to be taking a water sample at the point of ingredient. This is where we may expand and look at what we call heterotrophic plate count, where we'll be looking at all the bacteria as opposed to just a few in safe water, drinking water. Incoming water, so whether you're on a well or on city water, we want to be looking at how is it coming into our facility? Is it clean at that point? In the past, you used to be able to get away with just using municipalities' reports if you were on city water or public water sources. The way that many of the audits are going now, SQF-BRC, they are asking that you do a sample in-house to have that representative of what it looks like once it is in your building. Water and chilling and cooling systems, anywhere that is being circulated back around, or it could be a source of condensation. And then as we take these samples, ideally anywhere where we have water coming out of the wall or ceiling, we want to take the site at each one of those, not all at once necessarily, but we can rotate those sites to make sure that we collect all of them. Now, what many of you might be a little more familiar with is my drinking water, my water that I'm using, safe? And really when we do that, it's a presence absence of the coliform and E. coli test, commonly done with things like CoAlert or the indicator samples, where clear is good, there's no presence, and the change of color indicates to us that that presence is there.
All right, with the air and the water tested, we're going to talk about the surface. So the first surface residue that we can test for would be those tests that you might do commonly for your allergens or any chemical residue from the cells. So these are used on your daily basis to monitor effectiveness of your cleaning and your sanitation. And the pros to this is obviously you get those fast results. You can clean more if you need to, but it does have limitations. It does not verify that it is microbiologically clean. So you guys may be doing an ATP swab in your plants, and that's what that is. So that's checking for that surface residue. But with that limitation, we'd preferably test for clean to a microbiological level, and that's when we're going to check for the microorganisms on the surfaces. So what are we looking for? We're looking for those indicators and those pathogens. Why does this matter? Because it's going to evaluate the effectiveness of our cleaning and sanitation, but it's also going to identify those growth niches around the environment. Where are we getting growth? So the three areas that we're going to talk about for sampling is going to be the Rodak plate. So those Rodak plates are placed on flat surfaces that are impervious and are typically clean. So that's just going to be a set out plate that gets sent in for testing. Swabs are going to be used for your cracks and your corners, your crevices, your small equipment, anything that's small and you have to get it into a tight area for something that you want to test that's not easily gotten by a sponge. But lastly would be the sponges. So sponges are used for those large surfaces, the walls, the equipment panels, anything where you can get a really large area with that sponge and get a really good sample. The sponges you might see would be the cellulose sponge. The cellulose sponge is going to have a little more of a brown color. The negatives on these might be that, you know, with that wood pulp, it's going to have to be chemically treated before use. So there is some consistency concerns. There may be some substance that might inhibit growth. And also if it gets snagged or snarled, you might get some extraneous material into the sample as well. So more commonly, you are going to see that poly swab or sponge. So that's made from medical grade, man-made material. It's non-toxic, biocide free. It's really good. It's got an excellent survivability of the microorganisms. So as it gets shipped in, you know, it's going to really encourage that growth so that we can test it here at the lab. It's compatible with your leading tests. So any test that you are doing, it is compatible with. And it's also suitable for those abrasive materials, sharp edges, kind of things that you might get caught on the edges of tables, edges of knives and blades and stuff that might cut other sponges. So these will hold up pretty well to that. The other thing that you may have seen in your plant would be that 3M quick swab. So these are used when your testing is done in-house, but remember those should not be sent in for external testing. In those sponges and swabs, you are going to see that liquid. Remember that liquid is a neutralizing broth, and it is very important. So it's not just there to keep it damp, or that, you know, before your sample you should be pouring it out. You really want to keep that intact. It serves two purposes. Number one, it's going to neutralize anything that's got a sanitizer on it. So a lot of the samples that we take, we are going to take after the sanitizer application. We want to make sure that we're getting a test of the surface that we cleaned, not the sanitizer that we apply to it. So that it'll neutralize that sanitizer. And it's going to also maintain the viability of the microorganisms. So it's going to keep those things alive and ready to be tested before it gets sent in. So really important that we keep that broth in there. So now we're going to show another video about how to take a good environmental sample with a sponge.
In this section, we will cover sample collection for your environmental sample. Remember, the best results come from the best samples you can take in your plant and we can receive here at our lab. Remember, everything we're doing, that only contact with the sponge the things that you want to sample on the surfaces. So be very careful not to touch or adulterate the sponge with anything besides your desired testing area. So Matt is going to show us how to properly get a sample ready for the lab to test it for your environmental sample. First thing Matt is going to do with a black permanent magic marker, he is going to label the sponge, the outside of the bag of the sponge, with the date, description, and number matching what is written on the ARF. When your lab receives the ARF and the sample, we will match them up so that the proper test is done on the proper sample. Next, Matt will carefully remove the protective tab on the top of the bag. Only remove the tab when you are ready to test. Do not pre-remove them, as they can get dirt and debris into the bag that you don't want. So at point of use, remove the top tab. Next, he will pull the tabs on the open side to expose the stick. Carefully pushing up the sponge without putting his hand in the bag until the blue stick is on the outside of the bag. He will pull it up and out, making sure he does not touch the sponge to any surface he does not want to test. Please note, on the stick is a thumb stop, which is a plastic indentation. Never have your thumb or any other finger go past that mark to avoid contaminating the sponge. So now that he has it, he will go to his desired area and firmly press the sponge against the area, looking for that bend in the plastic, that he has enough pressure. So going in one direction, he will go back and forth with the head making full contact on the desired surface. So he's going to go back and forth until he has the complete surface. Now, he will flip the sponge over and go the opposite direction, rotating that sponge 90 degrees, so he's going the other way, ensuring that he gets all spots of his desired surface on both sides of his sponge. Next, we will have to return the bag, return the sponge to the bag for proper sealing. So he, without putting his hand in the bag and without touching the sides of the bag, will put the sponge back, grab it with his fingers on the outside of the bag, and twist counterclockwise to remove the stick. For proper shipping, make sure the stick is not in the bag. If the stick is left in the bag, it could puncture and also will cost the lab time, as they will simply have to remove it as well. So please remove before sending in. All the lab needs is the sponge. Now we want to make sure that sponge remains sterile. So Matt is going to roll the bag multiple times until it's secure, and then fold in the tabs to hold it tight. Remember, when you're sending in samples, most times you will have more than one sample. So you do not want the bags to leak. If the bags do leak, our teams cannot do the test as they have been contaminated. So we want those bags to be secure. When collecting samples, there's a few things that you want to make sure that you do not do to get the best sample. Number one, make sure you do not reach your hand into the bag to grab the stick. He has now had his hand completely into the bag, which nullifies that swab or sponge. Secondly, once you have the swab in your hand, make sure that you immediately use it. Do not walk around or talk to somebody or do something else and then come back. You want to avoid incidental contact, and you also want to avoid the swab being carried around in the air. So remove swab, immediately use. Make sure when returning the sponge or swab to the bag, you do not touch the top or outside of the bag and then return it. Remember, the outside of the bag is not a sterile environment just like anything else, so you want to return it directly to the inside of the bag for shipment. Next, when returning the stick to the bag, do not go past the thumb stop. Remember, your hands have been on the top of the stick all the way to the thumb stop. So you want to make sure that you do not go past that prior to taking the stick off. So above the thumb stop, twist or pull off, throw away, and then send the sponge for shipment. Lastly, when rolling up the bag, do not press it against your body and roll. You want to roll it in the air in front of you, not against your shirt or jacket. Doing these things will help you get the best sample sent to the lab for testing.
Okay, now that we have a good sample, let's get ready to send it to the lab. So how are we going to do that? Make sure the first thing is prior to packaging your swabs, check your bag for holes. If there is a hole in the bag, that's going to adulterate the sample, and we probably wouldn't test that. So you're going to get a call from your client services team letting you know if there's a hole in the bag. So check those bags prior to shipping. Remember to protect the sample integrity. Remove the sticks from your samples. Close those bags tight to prevent leaks. If there is leaking, again, some of the other samples might be affected. And then keep those samples refrigerated, 32 to 40 degrees Fahrenheit. But also remember, especially if you're using ice packs, that the swabs in the ice pack should not be in direct contact to avoid freezing. You don't want to freeze your sponges either. And then very important is make sure those arrive at your lab in a very timely manner. 24 hours or less is preferred, a 72-hour maximum. If you do exceed that 72 hours, that's another time you're going to get a call that, hey, your sponges or your swabs are out of time, and you got to make a decision on on what to do, and you probably have to re-swab and send them in again. So make sure that we get those in a timely fashion. Transporting them to the lab. Make sure you include your paperwork. We want to maintain that chain of custody. Make sure your analysis request form is in the box so we know what we're receiving when it comes in. Keep it cold during transport, that 32 to 40. Make sure it doesn't freeze, and then adjust your ice pack for seasonality, depending upon where you are, temperatures. Make sure you think about what's going to happen while that is in transit. And lastly is your shipping box or your cooler. You know, who is cleaning it? Is it staying clean? Does it need to be cleaned and sanitized or replaced if it's in poor shape? And then also, are your product and environmental samples being shipped together or separately, or are you doing something to separate those samples to keep them away from each other?
So now that we know how to sample, we need to understand when to sample. So it really breaks down into three major categories. The first is our verification activity, that our sanitation worked, right? Is our equipment clean? Are we safe to run? So you'll see at different facilities, different options here. As Robert had mentioned, what we like to see is that full representative sample, but we don't want to concentrate our sanitizer onto our sponge and falsely kill off the bacteria, giving us that false negative. So if you're doing after cleaning and sanitizing, make sure that you are avoiding puddles of sanitizer. Really let that dry and then take that sample. The alternative to that would be the sampling after cleaning but before sanitizing. Again, now you're missing part of your sanitation process. All these should be taken prior to operations. Now during production, we want to look at least 3 hours into our production run, and this is really starting to ask, are GMPs working? Are there issues with our raw ingredients that are introducing pathogens or bacteria to our line? A lot of times we'll see this at the end of a run as well, to kind of cap that off, to say our entire production run was successfully, we contained the biological risk during that time period. And the third is, did we maintain our food safety plan during disruptive events? And when I say disruptive events, I'm talking about construction periods, or if a piece of equipment goes down and it has to be completely torn apart by maintenance and put back together. The other element of that is, as your companies are growing or changing, there's going to be a lot of new equipment, new buildings. Before we start production in those areas, we want to take those samples and make sure that it's clean and ready for safe production of our food and products. So where to sample, we break it down into the 4 zones. And this is really to help mitigate our risk or do the risk assessment based on each of those zones. We know obviously stuff away from our production line will probably have a higher count. So the 4 zones that we start to use are zone 1, which is product contact surfaces, zone 2, which is in near proximity. So it could be our framework. It could be the food grade lubricants that we're using on our system, and other things very close to that production area. Zone 3 still probably in the production room, maybe a little bit outside, but a lot of the times this is where we start pulling in the things that are transient across production areas and non-production areas as well, such as the forklifts. And then finally, zone 4's, locker rooms, cafeterias, places outside of the production area. A lot of times our warehouses will fall into this as well, where we have closed product or store raw ingredients. When we break down where we're sampling, again, we kind of talked about using indicator organisms for zone 1. So if we have 100%, zones 2 and 3 are going to take up 75% of those samples. And then the other 25% for pathogen would fall into that zone 4 sampling. Zone 1 is going to be those indicator organisms, unless we see an issue or there's something that warrants the sampling of pathogens in zone 1. The meat industry has been pushing or doing this for years, where they've sampled pathogen zone 1. Thankfully, or not thankfully, but to this point within the dairy industry and the others, we get to use the indicator organisms. The thing to keep in mind, if you sample zone 1, you have to treat that as though you sampled product. So if there is a presumptive positive for Salmonella in our zone 1, FDA is going to look at that and say, it touched the product is now adulterated or contaminated.
Okay. So these sites that we're choosing, how do we go ahead and choose those sites? Remember, we're going to go out and we're trying to find these counts. We're trying to find these pathogens, right? So where are we looking to swab or sponge? Difficult to clean surfaces. So maybe communicate with your sanitation team. Where do we have problems cleaning? Where is it hard to clean? Where does it take us a lot of time to clean? Those are the areas that we should be hitting because they may show that something needs to change or something new needs on the sanitation team. Anything that is a critical location, areas between raw and finished, raw production and packaging, maybe changes in temperature. You go from a cooler area to a non-cooler area. So anytime that there's a change in location like that, anything that you've historically been associated with hits, anytime that you in the past have gotten hits or past people prior to you had hits. So areas that have shown a tradition of growth niches. Any areas exposed to moisture and or product residue. So if you're getting stuff that spills, if you're getting stuff that overflows, if you're getting stuff that, bags are breaking or stuff that's exposed to moisture and product residue throughout your plant. And just kind of in the back of your head, think something that is cold or is damp, I should be swabbing this for Listeria, especially as it gets closer to product contact. And then anything that's warm or moist or dry, I should be doing this for Salmonella. Especially think about things as it gets further away from your product contact into the warehouses and other maintenance areas. So we're going to go through a list. Again, this is not an all-encompassing list, but just things to think about as we're choosing sites in our areas to swabs. The floors and the drains. Anytime you see standing water, whether the water's from the outside, maybe the rain, or from the inside of condensation or dripping on your wash hoses, anything that is a ceiling or an overhead pipe, very hard to clean, very hard to get up on top of. Often scissors lifts are only brought in, periodically. So think about things that's over your head that's hard to clean. Anything that's going to refrigeration or condensation where you have a change in temperature. Wet insulation is something that if you see exposed insulation should be swabbed. Anytime you have cleaning tools, remember your cleaning tools are part of your environment. So sponges, brushes, squeegees, whether they're all, hanging on the floor or put away, are they being cleaned in between? So it's a good place to swab and look for things in between cleanings. Overhead rails, maintenance equipment that's being brought onto the floor, wooden pallets, especially wooden pallets that's on the move. From different areas. And then lastly, on this page, would be forklifts and pallet jacks. So those things that have those tires that are moving between different areas. Are you getting those tires? Are you getting the forks? Very important to swab those. Anything that has a recessed or a hollow object, so rollers or hollow table legs, switch boxes, box cutters, motor housings, things that may allow moisture in, but are not being cleaned. So think about, they're supposed to be watertight. Water's getting in there and your sanitation is not cleaning it because it is hollow. Anytime you see rust, anytime on equipment frames, pipes, shelving, metal grates, a lot of that older equipment is going to start to rust. So that's a great area that we should be hitting. Cracked or pitted rubber hoses, door seals, or walls. So if you can see outside through a door, right, you should be swabbing there because your door seal is broke. Your Strawman hoses, your cleaning hoses, if they become cracked, if they start to be in poor repair, is a great place to hit as well. And a little bit off the path is ice makers. Are you using ice in your facility? Obviously a change in temperature. Air filters, open bearings, wheels, on portable equipment or portable, tools that are being used, that are being wheeled in and out. Are we swabbing those wheels? And then lastly, light switches.
So when we talk about these sites that Robert just told us, we really understand where do we sample, but how do we pick that site directly? So let's look at the picture below and say that is a 10-foot by 10-foot picture. I'm going to throw up a poll here and tell me, where would you sample on this picture if you were told to go out and sample? So should be seeing the poll come up there. So I'm seeing a lot of B's, an A come in there, a couple A's. So ideally, when we look at this sample, thank you guys for your interaction there. When we look at this area, we want to be looking at how do we encompass everything. Obviously 10 foot by 10 foot, even for pathogen, is going to be too large of an area to sample the whole thing. I see a lot that there was a lot of bees. Did you step on the grate here? Was there moisture underneath? And what we want to identify there is that we're always taking the exact same sample between technicians. So if Robert goes out and I go out, we can sample that same area. And also, if there's an investigation that comes after it, we have enough description to be able to say, here we go, this is exactly where we took it. When we send into the lab, we want to be able to know that the sampling devices as well as the lab is doing their job correctly, that there's no additional contamination in the shipping process, in our process, in the lab's process. So we send a negative control. Ideally, this would be blind to the lab. So if you have sites 1 through 10, you can label it as site 11. Open the bag, remove the stick just as you normally would. Just never touch your sponge to anything outside of the bag. Close it back up and send it in for sample. We want to ensure that there is that sterility, no contamination, and we should see a negative control or a negative result when we have that test. When we get our results back, we want to be looking at them in a very timely manner. Obviously, as we talked about how fast bacteria grow, we want to be able to control that in the environment, and incubation periods only can be mitigated so far down. So it's really important when we get the results that we look at them immediately. When we have pathogens, or we can say negative, positive, plot them on the map, look at traffic patterns around that, where else we could be impacting. When we have our indicator organisms we can actually count, we can put that number in there. Now, the nice thing about today's society is that we have the technology no longer to have to put this on with pen and paper or with Excel spreadsheet even. There's actual environmental monitoring softwares out there that'll map out your entire facility. We have ideas around those that we'd be happy to share and work with you guys to get set up, but we'll take that as an offline conversation after the presentation. If we do have that positive, we want to do some investigational swabbing, or what we call vector swabbing. Now, there's no set distance from where we had that confirmed positive or out of spec that we need to go and look. It can only be within 10 feet of here. We want to take 360 degrees and look at where there might be harborage points, where there could be some of those other hot spots. Really, the idea of this is not to say there was a puddle of water here, I sampled the puddle of water and got a hit, we cleaned up the puddle of water. We want to be able to identify, where did that puddle of water come from? Was it a structural column? Did somebody walk through it and take it to a different area? And that's where we're looking for with these vector samples. Then finally, when we take a sample, we need to have some sort of record around it. So the important things is the date and time the sample is taken. So knowing what happened in your facility during that time period. As you see up there, product, what product was run, where we know the ingredients that would be a part of it. That detailed location so that we can go and repeat the sample or be able to investigate those samples further. Who sampled, so if we have to have that question. And then that date and the test that was requested of the laboratory. Again, going back to what Robert had said earlier, ideally within 24 hours that we want to be testing that sample, but no longer than 72. And that date and time would be that indicator. So I want to thank you guys for joining us today. We're going to stick around here with the full panel to answer any questions that you have from the presentation. But please, by all means, use our phone number and my email address, our website. We have some FAQ pages up there to help answer questions, but to continue on the conversation. And then as we answer questions, I'll leave this slide up. We do have some other webinars coming up here in the series that are showing on the screen. So if you found this enjoyable and informational, we invite you to register and join us for some of these other ones as well. So with that, thank you to the panel and thank you all for joining us, and we'll turn it over and open up the Q&A session as well as the chat function here to answer any questions that you have. So I see one question here about our air sampling, the 15 minutes. So 15 minutes is the standard when we look at the standard methods for the examination of dairy products, as well as the compendium for food. That is the given standard. The FDA recognized standard is 15 minutes is the minimal amount for collection of air samples. So Robert, do you want to touch on some of the environmental softwares that are out there?
Yeah, so we have a company that we work with that has environmental control software. Again, I think for the simplicity of getting it fit for what you need, I think if you could email or call Matt directly, and he can walk you through and show you a sample of how it works and what it looks like. So I think because for each plant, based on size and number of facilities, it's going to be very different. So if you could just go ahead and shoot him an email, he'll get back to you, and we can talk about what software that is and what it looks like.
So I see another one here about the yeast and wool Petri film versus plates for air quality monitoring. Everything has its purpose, and obviously within the right use. The advantage when we start talking about the plates versus the Petri film is obviously the amount of media and that is available for nutrients from that standpoint. Looking around the room, additional thoughts on that one? Both are viable, very viable options. And from our lab's perspective, we do see both of those come in for testing. So for the question that is, we have a lot of Listeria presumptives, but they all come back negative. Should we be worried about Listeria presumptives positive? Can I ask what platform do you know? Are you running that on a PCR where it'd be looking at the DNA, or Vitus type alpha platform would be looking at the protein antigen antibodies? And as we wait on that, I see the other one was, does ATP or test detect anaerobic bacteria? So ATP is actually, it stands for adenosine triphosphate, and that's actually going to be basically the energy cell of a protein that would be floating around in there. So any organic matter that is present on your equipment or where you took that sample is going to be represented on that ATP swab. Now, obviously, that accounts for much more than just bacteria, and that's why, as Robert had indicated during the presentation, it needs to be used coincident with the bacterial testing as well. Sure. So why I'd asked that question earlier about the Listeria presumptives coming back and not confirming out. If you're using a PCR platform where it's looking at the DNA, it is possible that's looking for all sorts of DNA. So there's what's called non-viable, non-cultural DNA, basically dead DNA that's still present and detected with the test. This may be what you're seeing as your presumptive result that then wouldn't confirm out after that. If you have more questions on that, again, that number up on the screen, or was up on the screen, by all means, I invite you to call in and we can have a further discussion as available there. Okay, so I see, do you have any question or recommended compressed air sampling procedures or recommended sources for designing a sufficient program? So really, if your compressed air is going to be used as part of the system that's going to be moving product along or in direct contact with the product, you'd want to be sampling that at the point of use, similar to what we had described during the water sources. Now, I know in dry environments, and Robert came from the chemical and sanitation standpoint, so feel free to add in here. From a cleaning perspective, when you use compressed air, you kind of blow everything all over the place. It might have clean on the surface, but we don't know where everything else. So people are trying to go away from the compressed air as part of their sanitation. If it is something like that, you can take more of a representative sample. So if you have an outlet where it'd be on the main line, excuse me, on the main line of your compressed air, after the compressor, after the filters, before the point of use, you could go and take that sample there. The thing to watch out for when you're taking compressed air samples is if you just take a Petri dish or a Petri film and blow it directly onto that, you're not necessarily separating out any bacteria that would be on there. So what you could see is one cell creating one colony. You could also see 100 cells that all went to the exact same point on your media that would also show up as one colony. So finding a way, there's compressed air adapters. Weber Scientific has a very affordable compressed air setup and air sampler that uses the Petri dishes with your target media that I would strongly recommend looking into. And what that's going to do is it's going to cycle that compressed air in there and kind of break the cells apart from one another, giving you a much more representative sample of what's in your compressed air, if anything. I guess the last thing to touch on is compressed air wise, a lot of times you'll see yeast or your mold as being your big contaminant in compressed air lines due to condensation and that presence of moisture. So I see a question here of, do you have any suggestions on the type of environmental sites to test for EB? So when we start talking about EB, we said that that's going to be one of the indicators for our coliforms and our Salmonella. So similar to what we had described as sites that you would be testing for your Salmonella, that dry or moist, a little more heated areas, or if you see that contaminated risk for Salmonella as a presence in your product or raw ingredients, that's where I'd really use that EB test. Now, excuse me, this is kind of in lieu of your Salmonella test as presence absence and gives you that longer indication. So we have seen EB tests across all 4 sampling zones, but this is where you really want to put that in place on zone 1, where you'd be utilizing, so that product contact surfaces, where you'd be utilizing your EB testing. Again, be happy to have a further conversation. It's a little hard to answer a pinpoint crack or crevice without knowing exactly what facility you're in. And that's just, something I'd be happy to discuss with you, or our team would be happy to discuss with you outside of here as well. So see a question here. We currently sample Salmonella in our environment. Do you recommend that we consider EB sampling as indicator instead, or continue with Salmonella sampling in the environment? Again, as a dry dairy powder facility, I would say where your product is actually touching, you want to have some sort of idea of what you have for a bacterial load there. So I would recommend taking some samples and building a baseline study around your product contact surfaces with that EB as the option. So the buffer solution, the question here is, do we need to wipe off the buffer solution in the blue sponge swab after swabbing the surface of the equipment? Quick answer to that is no, it is not a requirement. There's not really enough there in the way it dries out to have nutrient that would promote the growth of bacteria. I know that's typically one of the concerns that arises. The other part to know about the buffer solutions is all the buffers, the neutralizing broths, because neutralizing buffer is one of the neutralizing broths. All those are on what's called the generally recognized as safe list for food environments, or GRAS list for short. Any other questions that I missed on there or that are arising?
Like Matt had said, if you guys do have anything, we encourage you to email or call, and your specific situations are always different across all factories and all industries. We'd love to, talk to you about that. So please don't hesitate to reach out to Matt via phone or email. We obviously appreciate everybody being here, and hope to see you at the next one, next webinar. And we appreciate everybody's time and attention.
Yeah, and just to touch on that, the next webinar will be again at 10 a.m. Central Standard Time on September 16th. And that's going to be diving in a little bit deeper to choosing your indicators versus your pathogens and really how they're growing, where you'd be looking for those. So some of those questions that arose around that, that would be an additional webinar source to use. All right. Again, thank you all, and we hope to see you then.